We are trying for the first time to install a little piece of software today. So this is the first time that I just publish it. So there's going to be a lot of problems. So a couple of disclaimers. Installation should work out of the package if you already have PyEMMA, MDTrash, Scalar, and the visualizer software installed, which might be the case in some installation. Okay? A second thing that I want to point out before we start getting questions is there exists a known issue. Okay, so we have a successful installation. So, welcome to Projection Explorer. So Projection Explorer actually is something that in one way or another every one of us has done before. It's just in different ways. You have an MD simulation and you get some clusters, and perhaps GROMACS does the clustering for you and then writes the frames in some file, and then you have to use that file and go somewhere and then visualize those frames on your favorite molecular visualizer, BMD, PyMol, whatever, okay? So we're always having this disseminated information because we are abstracting, right? We are turning molecules into numbers, numbers into dimensions, dimensions into projections, projections into discrete states into a matrix into vectors, and then we're like, okay, so where is my molecule? Like, what does it mean now when I show you the free energy surface? What's behind the minima, and what do the axes actually mean? Okay, so this is a question that most of us are all the time asking, and as soon as you're a little bit savvy, you really know your way around it. You use GROMACS or you use BMD or whatever. So this is a first attempt to try to unify all of that in a notebook, okay? So the module, projX, has two submodules, visualize and generate, okay? And the most important one is visualize, because it gives you an interface to mix projection information with molecular structure information interactively. We will see the interactivity or interactiveness in a moment, okay? There's another module, generate, which somehow contains the tools that help visualize work, okay? But in principle, we should go with visualize, okay? I would not be able to do anything if it weren't for this Ngl module. I think we should give a lot of credit to Alexandra Rose, which is the one that wrote it in the first place. Actually, the PDB is now displaying by default all of its structures. If you go to the PDB databank and you click on a structure and you want to have it displayed 3D immediately, the PDB databank is actually using this guy's code. So this is a really good code. What I have done is just a little bit of connecting here and there, okay? Of course, we're incredibly grateful to our friends of the Pande group that have written MDTrash and, to PyEMMA. Good. So, how to use issues, documentation. How to use Projection Explorer. Let me go to the notebook that I should have opened here. Okay, here we go. Again, the familiar by now notebook structure. We start by importing projX, and we start by loading the molecular information in the familiar way of a trajectory and a topology, okay? Now, please, those of you who have the notebook running, try to execute the cell number 3. If you have never done it before, it will compute a Tika projection and project the data, the BPTI data. If you've run it more than once, it will just skip it and load the data, okay? And now, we are going to use projX visualize free energy surface, where the input is just the trajectory file, the topology file, and the projected trajectory, okay? So this is something that we generated with GROMACS. This is just a structure, and this is our projection. So this is the part that you actually want to understand. This is the exotic Tika, or kinetic Tika, or PCA, or whatever, okay? And Projection Explorer is gonna do some stuff and provide you with this beauty, okay? So this is the Ngl widget. It's a widget to visualize molecular structures inside of the notebook It has a lot of options. You can tell it to provide you with a graphical user interface, okay, which is here, where you can change things about this molecular representation. Okay, so this is the BPTI notebook. And the cool thing about all of this, if I manage not to break my neck in trying, is that now the upper and the lower plots are connected. So that if I want to know what this minimum actually is, I just have to click on it. And it will show me the molecular structure behind this minimum. Or if I want to click here, okay, it will go to that point in the potential energy surface and the free energy surface. Or I want to explore this minimum here, right? So clearly there is a lot of room for customization here. You can change the molecular representation, you can highlight given residues. But in principle, it answers a very fundamental question, which is what does this block mean and what does this block mean, okay? Yeah.
But it's, I mean, there are many structures there, so it just chooses one that is closer to the...
Exactly, so this is like how it works. This is a projection. The fact that this part of the projection space is displayed blue is, as Joao has just said, is because there are a lot of structures that occupy this position in space, okay? So there are a number of them to pick from. How I choose which one to display is another question. Yeah. This is which has been answered, and we can talk about it. But this is so the first functionality. So this is just like how it actually started, because somebody who's present in the room, but I won't mention why, just asked me like, how do I know what this means? Like I want to like click there and see what happens. And I said, well, I know a way around it. You just have to do 3 things, and then there's the structures. But it seemed like complicated for people that perhaps have problems installing software or working in 2 different environments or changing program. So this is why we put this thing together, okay? The other nice thing is that you can actually see it in a time-resolved manner, okay? So we can actually choose not to represent the free energy surface, but just to show the trajectory data. And again, we'll have the familiar, okay? This, you should recognize this trajectory already, the thick projections. And again, so this is what's happening, okay? So I see where I am here. This little red dot is telling me where I am, and I'm seeing the trajectory. But it works interactively, so I don't have to wait until I arrive there. I just can go like, okay, so what happened here? And the trajectory will be displaced to exactly where that point in trajectory space is. So this provides a very intuitive way of interacting with it. This is this other method, which is called visualize trajectory. Okay, I want to see the sequence of events, not necessarily the free energy surface where the time degree of freedom has been integrated out. And this is just, let me see if I can get it a little bit smaller. So I just want to brag a little bit how all of these 3 plots are now connected. Okay, so this is just both functionalities plugged in together across all 3 plots. So if I click here, the trajectory moved to a point that represents this point, but also the structure. And now to come back to your question, Joao, when I click here, the trajectory could in principle show me any point between here and here and between here and here. Right? So any one of them is actually equally good.
If you click one little bit to the side, probably it will show you different points.
Exactly. I can actually just click again and see where it goes. Yeah. Okay? But this is the right way of thinking about this, right? Because all of these points have been projected to this minimum, okay? So this is kind of like click and view. This is the generate part, okay? So it gives you the method sample, which will generate the objects that are needed to use the visualization. I put this in the notebook so that everybody sees how it's working internally, okay? So I can just generate a sample starting from a trajectory, a topology, and the data, and it will provide me with the positions. So it will provide me with a sample, okay? So geometry is this MD trajectory here. I asked for 200 points and I got 191. This is just a little bit so that the algorithm doesn't iterate the clustering algorithm too long. You don't really care if you have 200 or 191. So these are the frames that are being plugged into the representation, and these would be their positions here. Data samples, okay, 191 times 2, okay? And this is where the linking is actually happening, okay? I can create a plot independently, okay? So these lines here don't have to do anything with Projection Explorer. This is how you would just plot the free energy surface. And then I can get a widget that is connecting the data sample that I just generated here, okay? So upstairs I generated these objects and these objects, and they are being fed in here and here to the current axis, okay? So this axis could have a contour plot, or could have anything else, or couldn't have anything, okay? I'm gonna just get the same thing again. Okay, now I have chosen to display the cluster centers onto which I am clustering. So again, the same game. I click somewhere and it goes to the closest point. Click here, it should go to this one. Okay? And this is where it gets, to me, also interesting, okay? Because so far, we are still on a very click and see, okay? So you click and you see. This is almost equivalent to storing the geometry and opening somewhere else and then like write a little paragraph. Okay, in this conformation, the outer loop is pointing towards the binding pocket or whatever. So this is why we have a method that's called projection paths. And projection paths is an attempt at empirically determining what your projection actually means, okay? I am being a bit careful about how I phrase this because this is a post-hoc analysis method, okay? I won't go into how did we arrive at the projection because that's another problem, a difficult one, and a thing of itself. This is just, okay, provided I have already projected onto something, what does it actually mean to move along that coordinate back and forth? So for that, we generate projection paths, okay? And these projection paths contain geometries that move across the projection space, advancing in one coordinate while keeping all the other coordinates as quiet as possible, so that you're only seeing the conformational changes associated to an increase in the value of the coordinate that you want to sample. When you see the plot, it will immediately be clear what is meant, okay? So we generate a dictionary and paths and the data that's associated with it. We can look at this later with more time. And I have chosen to sample, okay? Where's the working? Oops. Aha. I path is not defined. Because I didn't execute this. Okay, so I think the concept is now much clearer, which is the geometries that are being shown in the widget lie all on top of this line. Okay, the green line. So what I have done is I have gone back to the data. I have started somewhere in space and I have said, and now show me a structure where coordinate number 2 has increased and everything else has stayed as quiet as possible. This is why this line, while it is climbing up the sample coordinate, is not really diffusing in any other, okay? And those structures are displayed up here, okay? And now, because I know this system, I know that the second tika is actually something happening. I don't remember if it's this loop or this loop here, but as we move along and we see the point, okay, climbing here, you will see that what's actually, or you will not see, that what's actually happening between this minimum and this minimum, okay, it looks like it's a movement here, but this is part of Joao's question. It's because when we sample coordinates, it's impossible to keep the entire structure quiet, but it's actually something happening there. There are two types of paths that one can sample now. One is a path that is forced to sample the coordinate while minimizing diffusion in all of the other orthogonal ticks, and another one that's called the minimum displacement path, and the other one, it's a path where what you do is you minimize the RMSD between adjacent structures. So you are still enforcing the fact that I want to walk along the line of projection number 2, but you are giving more flexibility as to what structures you choose to display so that the movie that you see in the end is smoother, okay? So now you will notice this, hopefully, and what will happen is the path in coordinate space is a bit jerkier, I think. I don't know how to call this path. A little bit more broken because I have relaxed how much my path can diffuse in coordinate space. But the movie should be a bit more smooth because now I am minimizing RMSD, okay, along coordinates. So let me see. Yeah, it's a bit hard to see. You have to know the system to see what's going on, but you see my point, okay? I can go see here and see what happened. I can also change the coordinate. I chose the coordinate number 1, but I have actually also the coordinate number 2. And the coordinate number 2, it's this one, okay? So this path looks pretty bad, okay? It looks pretty bad, but it's actually doing what we have asked from the path, which is the path starts here and always advances along the sample coordinate. It diffuses, okay, on the orthogonal degrees of freedom, but that's not really that bad because what we want to see is what happens when you move along this axis, okay? And now the figure should actually show clearly the opening of, let me see if I'm looking at this correctly. It's this loop here. This loop here will clap to the insides. There you go. Okay, so this is the first process in the BPTI. This is when we see the coordinates up here. Okay, here you can see nicely what's happening in this jump is that this loop is flapping. So here it's there, and now I think I'm reading this correctly. This loop should flap. There you go, and then after that we go back, and it's back at its original position. One moment. I can go back to the, not the mean RMSD, which is the visually smoother but more diffusive, or go back to the minimally displacing path. And now the line in the plot, well, still looks, it doesn't look as straight as the other one, but it's pretty much a straight line, and it conserves the minimally diffusive. You can also provide your own clustering objects. I think this is also a nice feature. Okay, so we're just doing pure PyEMMA here and getting a clustering object, and we're plugging this clustering object here back into the sample generating method. And now we can take the sample data and the sample geometries, link this with, okay, so I want you to pay a little bit of attention to the fact that here I am plotting the cluster centers and here I am plotting the contour, okay? This is all of the flexibility of the matplotlib library combined with the linked widget, okay? So the plots can actually have whatever you want them to have. And, you know, here we are. So the red dot or this dot or whatever. So I have really tried to keep it nice and documented. The GitHub repository is still closed because there are some things that I want to figure out, but you can build your own documentation. And I have also put a copy of the HTML documentation on my webpage. But all of the methods are documented the way that you would expect a package like this to be documented. And as you see, there are some issues with the installation, but half of the people got it installed. So for me, that's already a success. So I hope you like it, and thank you for your attention. So there was a question somewhere.
Yeah.
Yes.
So basically, if you look at one frame of the shown of the molecular structure, that is 1 point linear, right?
Sorry, what?
So one